Welcome to Unit 5, Lecture 3. In this lecture, we're going to talk about cell transport. So last lecture, we talked about the membrane. We talked about the structure of the membrane. And if you remember, the membrane is selectively permeable. So what that selective permeability is the fact that it lets some things through, but not everything. It's the gatekeeper for the cell. It's what helps keep the cell intact, keep the cell healthy. So we're going to talk about how it's selectively permeable in this lecture. Learning objectives. Explain how passive transport works. Explain how active transport works. There are two main types of transport. One, passive transport. Two, active transport. So passive transport is simply going from an area of what's known as high concentration. So you can see there are a lot more molecules in the blue here to an area of low concentration. Okay, so you can see there's a lot less of them. So what this basically is, it's just like going down a slide. Okay, so where it's higher, right, it moves to where it's lower. Okay, and there are, well, here we're going to talk about a third type, but there are two main types, or I guess I should say, there's one basic type, and then it's got a little bit of tweaks to it. Okay, so diffusion is basically just going from an area of high concentration here to an area of where there's less. Okay, just right through the membrane. Oxygen does this, CO2 does this. Okay, just travels right through the membrane. But then there's the facilitated diffusion. What is peer facilitators? They help you do stuff. So facilitated just means help. And that help comes in the form of a protein. All right, I like to call this the easy pass diffusion, type of diffusion, because what happens is, whatever reason, these other molecules, the triangles, okay, and the blue circles, can't make it through the membrane by itself. Some of that is that they're too big. Okay, so glucose needs to do facilitated diffusion. Right, glucose needs to do facilitated diffusion. It's too big to get through the membrane by itself, so it needs a channel. It's basically a special lane for that molecule to get through. That's why I call it the easy pass. Okay, then the other type of transport is active transport. Active transport requires energy. Okay, it requires energy. It requires that ATP molecule that we made through cell respiration. So this is part of that metabolism. Remember, metabolism is every single chemical reaction that is happening in your body at any given time. And so we need ATP to make this type of transport happen. So if you'll notice here, this is one version that we're going to talk a little bit more about in the next couple of slides. But here, these are going against the concentration gradient. So we're going from these, an area, so you can see these little orange diamonds, okay? We're actually trying to push more of them this way, all right, where there's actually more of them already in the cell. So this is the high concentration, this is the low concentration, and we're actually trying to force them to go that way, okay? So it's kind of like pushing you up the slide, all right, whereas where passive transport is basically just going down the slide, okay, down the slide, okay? Active transport is actually trying to push you up the slide, right? Another type of passive transport, and it's really no different, it's just the fact that we give it a special name, okay? Osmosis, which is the diffusion of water. It's just a name for the diffusion of water. And so what happens is that you can see in this tube, okay, we started out with sugar molecules and water, sugar and water. So we have a higher concentration of water over here, a lower concentration of water over here. So over the course of time, what happens is the water actually moves to the other side of the tube. Okay, once again, it's diffusion. So it's going from a high concentration to a low concentration. In this case, we're just talking about the movement of water, right? That's it. All right, so going from high to low. Right. Osmotic regulation, okay, or osmoregulation, is really important for cells. This is why you need to stay hydrated, okay? But if you are too hydrated, this could be a problem. And if you're dehydrated, this could also be a problem. So there are three types of solutions when we talk about osmoregulation. There is hypotonic, isotonic, and hypertonic. Okay? Hypotonic is when there's more water outside, so there's a higher concentration of water outside and a lower concentration of water inside. So the water is actually flowing into the cell. Okay? It's going into the cell. Now, if it happens too much or too quickly, okay, you can see here the cell actually bursts. The cell actually bursts for an animal cell. In a plant cell, however, because if you remember, they have that cell wall, right? They actually don't burst. They just kind of swell up a little bit. This is called turgid. Now, this is where plants actually prefer to be. Plants actually prefer to be turgid because that's pushing out against their cell walls. That's helping them stand upright and be okay. All right. Whereas for animal cells, without that cell wall, they'd basically pop like a balloon. So I like the O in hypotonic. Whoops. The O in hypotonic looks like a swollen or bursting cell. Okay. Isotonic, just like iso means the same in geometry, okay, if you have an isosceles triangle or whatever, iso means that the concentration is equal. All right. Okay, so what happens is water's moving in and water's moving out kind of at the same rate. Now, if you remember, you may have heard the term equilibrium before. Okay, equilibrium just means equal. Equilibrium. So the solution is equal on both sides. So water is just kind of moving in both directions. Now the thing about molecules is molecules never really stop moving unless they're frozen, okay, and they become a solid. But in liquid form, molecules and gas form, molecules are just kind of bouncing around all over the place. So they're going to come in, they're going to go out, but they're going to do this at the same rate. Okay. Now this is where a plant, okay, it's not terrible for a plant, but this is where you start to see the plant shrivel up. Okay. That's where they start to kind of like shrivel up on themselves. Okay, and they start to wilt. So if you just water them a little bit, then they kind of pop back up into this turgid state. Okay, and last but not least is hypertonic. So in hypertonic, okay, there's a lower concentration of water outside and a higher concentration inside. So what's this going to do? It's going to leave the cell. And the same, like the O kind of looks like a swollen cell. All right, the E kind of, the E kind of looks like it's starting to shrivel up. All right, and so in a hypertonic solution, the water's leaving, causing the cell to shrivel. All right, and you can see that happening also in a plant cell. The plant cells don't, again, because of that cell wall, don't necessarily shrivel the same way that an animal cell does, but it's still the same kind of method. And like I said, this is where you'll start to see plants wilting, okay, and they kind of start like falling over, and their leaves kind of get a little like, what's the word? A little wrinkly and stuff like that. So you just add water to them, and they go whoo, and they pop back up. Okay, so remember, osmosis is just the movement of water, the diffusion of water from an area of high concentration to low concentration, but it's really super important for cells. Okay, active transport. So remember, with active transport, we're using ATP in some way. So there's a couple of different ways that we can do that. So the protein pump, which was what was in the example of active transport on the first slide. This is where we're basically just using a protein to move things against the concentration gradient. Okay? All right, so we're just trying to move things from one spot to the next. All right, then there's endocytosis. So this is where we're bringing some molecule into the cell, but we have to actually enclose it in a vesicle and move that vesicle. Okay, so we're bringing, endo means in, cytosis means cell. So we're bringing that into the cell, but we have to wrap it in this vesicle. All right. And then there's exocytosis, which is the opposite of endocytosis. So exo means out, and cell. Okay, so we're taking that vesicle and we're shuttling it to the cell membrane so we can expel the molecule, whatever it might be. So this process of moving vesicles, it uses ATP, okay, because the molecules can't do it by themselves. So this is part of your metabolism, is shuttling these little vesicles around your cells and taking them to wherever they need to go, whether it's into the cell through endocytosis, whether it's out of the cell through exocytosis. Right, so here you can kind of see this cell right here. All right, this is one cell. Okay, it's starting to actually try and wrap around this, whatever this happens to be. Looks like a bacteria or something like that. I'm not even sure. Okay, and it's trying to actually wrap around that cell so it can kind of suck it up and do whatever it needs to do with it. So it's going to make a vesicle around this cell, okay, all right, to try and do something with it. Probably eat it. That's probably what it's trying to do. All right, so I will see you in class. Have a great day.